hopefully that's okay. Great. So my name is Robyn Bell, as I've just said, and today I'm going to talk about a gene therapy approach to TTP. So just to introduce where I'm from. So I'm from Imperial College London, and we are based in South Kensington by the museum, so the Natural History Museum and the Science Museum, which is very nice. And this picture here is my research team. So we're called the Gene Therapy Group, and we are within the National Heart and Lung Institute at Imperial College London. So I just completed my PhD, and they very kindly offered me to stay on as a postdoc. So postdoc is just post your PhD. You can stay on as a researcher, and it's sort of the position after a PhD. So I apologise if this is oversimplified. I just wanted to assume a baseline knowledge, and then we could build from there. But I'm sorry if this is boring. You can zone out for a bit and zone back in if it's an interesting point. So obviously we have ADAMTS13, which is we refer to as a protease. So it has enzymatic activity, and it is able to cut up other proteins. And that other protein is von Willebrand factor. So the only known substrate, or the only known sort of object of ADAMTS13 is von Willebrand factor. And what it does is ADAMTS13 is really good at cutting up really long lengths of VWF into smaller pieces. So this is the sort of normal healthy activity of ADAMTS13. When there is an ADAMTS13 deficiency, and obviously this can be caused in two ways, I'll come on to that. Unfortunately, because the enzyme is not there, there's a deficiency, this VWF strings are not cut up into smaller pieces, and they remain very long strings. So if we sort of look at this in a more realistic way, so here's a schematic of a blood vessel. So this is the lining cells of a blood vessel, which are called endothelial cells. And within your blood vessel, we have platelets, as you will be very familiar with, red blood cells, and we have VWF protein. So hemostasis, in normal hemostasis, and by hemostasis I mean the process of stopping bleeding, we have a scenario where there may be an injury, so disruption to your blood vessel, a cut, or there may be the scenario of inflammation, that there is a trigger, and this leads to exposure of a protein called collagen, which is underneath your blood vessels. And when this is exposed, von Willebrand factor is very good at sticking to this. I like to think of von Willebrand factor as a really sticky string, and it becomes elongated. And when it is in this elongated form, we get lots of sticky platelets sticking to it, and this leads to this clot formation. And in normal hemostasis, so when we have a cut, we want this to happen. We want this clotting to stop, uncontrolled bleeding. What ADAMTS13 is required for is then to resolve this clot, so to break it up so that it no longer is a clot within when the blood vessel has been healed. So ADAMTS13 chops it off and it resolves this clot. So now let's look at the scenario in TTP. Just to say that the function of ADAMTS13 here is to reduce the size of VWF and to minimize the formation of clots. So when we look at the scenario in TTP, again, there's this injury or there's a trigger event and exposure of the collagen underneath the blood vessel. Again, VWF will bind to it and it becomes unraveled to this really long string, which is very sticky, and you lead to this clot formation. The difference here is that in patients with TTP, there is a deficiency of ADAMTS13 and the clot is not resolved, so it's not broken down. And therefore, we get something called narrowing of your blood vessel, and as red blood cells pass through, you see this fragmentation of red blood cells, so they sort of burst open and they become fragmented, and this is called schistocytes, and this is often something that can be found in the blood of patients with TTP. Also, these clots can become dislodged and they can travel to very small vessels, often in the kidney and the brain, and then lead to organ sort of lack of blood flow to organs. I'm sure this is maybe very obvious to a lot of you. I just wanted to introduce it. And as a consequence of the lack of ADAMTS13, the VWF is not reduced in size and the clot formation is not minimized. So as you all are aware, the forms of TTP are congenital, where the ADAMTS13 gene has an error in it. So we call that a mutation. And this is the minority of patients with TTP have congenital TTP, but still a form of TTP. And as a consequence of this error in the gene, either the protein won't be made at all, so there'll be none of it, or it's made but it doesn't have the correct function, or there's very low levels of it. And there's different mutations in different patients. But the end problem is that VWF is not cleaved and the clots are not minimized. Again, if we look at immune-related TTP, in many cases, the gene is fine, the ADAMTS13 gene is fine. However, the immune system of that patient starts to recognize ADAMTS13, and these are the self-antibodies I'm sure you're very aware of. But the end result is the same in that VWF is uncleaved and remains these really long strings. So treatments of TTP have the main goals of either trying to boost ADAMTS13, so plasma infusion, trying to reduce the amount of the antibodies, so this can be through corticosteroids or rituximab or plasma exchange, and more recently the goal of trying to prevent platelets from binding to VWF through the use of caplacimab. Probably not saying that very well. But these are sort of the current treatments with the main goals of as what I've just said. More recently, recombinant ADAMTS13 is being investigated, and I'm sure Professor Scully spoke about this, with the idea that you inject the protein, ADAMTS13 protein, that is being made in a outside of the body. So we call that recombinant ADAMTS13. Infusion of this protein has the end goal of boosting ADAMTS13 levels in the patient. And as I'm sure you're aware, the phase 1 trial for this recombinant ADAMTS13 was completed, and phase 3 is ongoing for congenital TTP, but also phase 2 ongoing for immune-related TTP. And that's really the, I'm sure Professor Scully was giving you the expertise there. But I think when we look at recombinant protein therapy, not just for TTP, but say for haemophilia, there are some disadvantages of this sort of therapy. One being the frequent nature, in that you have to re-administer this therapy. Another being the difficulties associated with producing it. And the associated costs of administering this treatment regularly. So not saying that this is a bad therapy at all. I think just as scientists, you're always looking for alternative treatments. And one such alternative treatment, which is what I'd like to speak to you about and what my PhD was about, was potentially trying to answer this question: is gene therapy a suitable treatment for TTP as a novel therapy, and that was basically the subject of my PhD. So just to explain how we can apply gene therapy and what gene therapy is. So regardless of the cause, so either ADAMTS13, a congenital problem, or a immune-related TTP, the aim of gene therapy is to try and treat it by delivering patients a healthy copy of the ADAMTS13 gene. So sounds simple, but the problem is how do we then deliver this gene into patients? So the ultimate goal is if we can deliver the healthy copy of the ADAMTS13 gene, this gene will hopefully be able to produce lots of the ADAMTS13 protein within patients, with the hope that you raise the levels and you can see an improvement. But the biggest problem that we have in the field of gene therapy is delivery. How do you get the healthy genes into patients? So what you need to do is disguise it. So if you were to deliver genes directly into patients, it would be broken up very quickly and it wouldn't have any use at all. It would be pointless. So what we need to do is protect it and almost disguise it from the body, so the body doesn't then recognize it as foreign. So there's multiple ways that we can do this. So here I've just shown a diagram of if we surround the healthy gene with fat. So this is almost like a fat globule. And this is one way of getting the genes into the body. Another way, and this is the way that I've been investigating, is to use viruses. So you guys will be very familiar that COVID-19 is a virus, and COVID-19 is extremely efficient at entering the body. And what we want to do with gene therapy is harness viruses for what they're good at, which is getting into cells, getting into the body, but we don't want any of the disease associated with it. So what we can do with viruses is we strip it of all the bad things that it does. So we gut it, but we keep the function that allows it to get into the body. So we take the good bits and we get rid of the bad bits. And this is the basis of using viruses as a gene therapy vehicle. So this is one sort of illustration of a virus, but there are many different viruses out there that we could use. We can harness them as delivery tools. So when we have these options available to us as a scientist, you've got to go through the pros and cons of the different systems. And some of the considerations that we have is, where are we trying to target? Which part of the body are we trying to deliver the gene to? Is it the lungs? Is it the liver? And that really depends on where this protein has its function. So with ADAMTS13, it works within the blood system, obviously. So that's something we have to consider when we are designing a gene therapy. Another consideration is, how big is the gene that you're trying to deliver? So ADAMTS13 is very large for a gene. It's considered one of the larger that you're trying to deliver to patients. So that then limits which viruses that you can use as your delivery vehicle. Another consideration is, how long would you like your protein expressed for in the patient? So would you like it, is it a chronic disease? Does it therefore need, do we need it produced for the lifetime of the patient? Or would you like a short burst rather than a longer duration? Another consideration is, do we need to administer this therapy multiple times or just once within the lifetime? And finally, sorry, my thing's in the way, I can't see what that says. What's it say? Sorry, it begins with S. Sorry, my strip's in the way, I can't see what it says. The final words.
It says safety and toxicity.
Thank you.
Sorry,
yeah, so different viruses or lipids have different safety considerations. So of course, they are foreign. So anytime you put something foreign within the body, it might be recognized by the immune system. So these are all the considerations we've got to think about when you design a gene therapy. And the reason that my PhD was based upon this particular virus is because of some of the reasons I'm going to hope to explain to you. So one of the other considerations when you're trying to design a gene therapy for TTP is, how is it best to administer? So you may think the most obvious route of administration would be through intravenous, so through the bloodstream, with the idea that if you deliver the virus through the bloodstream, it may reach the liver, for instance, and you might get production of ADAMTS13 within the liver. The end goal is that this protein is then distributed around the body through the circulation, because that's where it has its function. So I think for TTP, the most obvious route would be delivery through the bloodstream. But an alternative delivery route, and the route that I looked at, was what about if you inhale the virus, so deliver to the lungs? And you may think, why on earth would you ever target the lungs to deliver a gene therapy product? And it's because the lungs are full of blood vessels. So actually, they're very good at getting viruses from the lungs into the bloodstream to then go around the body. So during my PhD, one of the questions I looked at was, what if we deliver the virus intravenously, and what if we deliver it via inhalation? And then we didn't know the answer before we started of what might be better and what might be worse, but it was just one of the sort of questions I had within my PhD. So when we look at some of the advantages and disadvantages of both systems, obviously, if you deliver intravenously, it's quite non-invasive. You can get the protein into the bloodstream, which is exactly where we want it to be. However, a negative of this delivery through the bloodstream is that it then is easier for the immune system to recognize it. And I found in my PhD that we were able to deliver the gene therapy once via intravenous administration. However, we couldn't deliver it repeatedly. And that's because the immune system clocked onto it after one dose, and it was able to recognize it when you then gave it a second time. However, an advantage of if you inhaled the gene therapy was that you could repeat administer it. So we can give it multiple times, and the immune system does not put up a fight against it. And that really is one of the major benefits of delivering this gene therapy that we've produced in our lab through the lungs, is that you can administer it multiple times. And when you have diseases that are chronic, so they're lifelong diseases, that's a major advantage if you're able to repeatedly give it. Another advantage of delivering to the lungs is that it's then confined to one organ. So the reason the immune system is able to pick up the virus when you deliver it to the blood is because it goes to many different organs. And actually, you may want to just have a sort of safety in mind and think, I'd rather deliver it just to one organ rather than distributing it all over the body. A potential negative of inhaling the gene therapy is whether we will achieve sufficient levels of ADAMTS13 in the bloodstream. And that's something that may be addressed further down the line, but it's whether dose could come into that, how much you give, and things like that. So I think many of you might think, well, how does something like gene therapy apply to acquired TTP, in which the gene is absolutely fine in a lot of cases, but it's an immune response to the protein? And there's sort of two ways of looking at this. Either you produce enough of the protein that you saturate, so you sort of outcompete the antibodies within the patient, or an alternative way of going about this is that you slightly modify the ADAMTS13 protein so that the antibodies can no longer recognize it. So then you don't get this response from the patient's own immune system against the protein. And this is something that down the line would need to explore both and see which is the best strategy moving forward. So just to say that we're not the only ones looking at gene therapy for TTP. There are multiple labs around the world looking at it. And the difference between our approach and other people's approaches, is that another tactic that's taken by another lab is that instead of delivering the virus, so the gene therapy product, you deliver platelets that have been modified so that they can produce ADAMTS13 themselves. So this would be a regular infusion of platelets, and the platelets themselves generate the ADAMTS13 to work in the body. So that's another approach looked at by other research groups. Other labs are looking at using different viruses. So I said previously that there's not just one virus available in the world of gene therapy. There are multiple, and some are just choosing to look at using different viruses. And I just want to sort of clarify the steps that are required when you are producing something new like a gene therapy for TTP, because I think it's important to realize where we are in this long process that is required to generate a new therapy. So it first starts with an idea. So can we apply gene therapy to TTP? You then got to design a therapy towards TTP. So all those factors that I spoke about earlier, such as what's the best virus to use, where are you going to deliver it to? These are all things that need to be ironed out quite early on. You also need to develop ways to test if your therapy is effective or not. So it's only one thing delivering the therapy, but how do we know if it's working well or not? And that's called sort of assay development. It's a way of basically us knowing if we are able to deliver ADAMTS13 successfully or not. Another sort of process in a step in this process is finding models. So we can't deliver these new therapies directly to humans in the first instance. It wouldn't be ethical, and it's very unsafe. So we first have to test our new therapies in animal models. And unfortunately, this is still a part of the sort of therapy process, is using animals. And hopefully in the future, it wouldn't be if you can find a replacement. But at the moment, it is still a necessary part of drug development. And unfortunately, the models available for TTP, so the animals models of TTP, are quite tricky and not perfect ways of testing therapies. So these are all things that need to be ironed out when you're developing a new gene therapy. And once you've got to the point where you've found a product that you're happy with, the next hurdle is being able to produce it. So producing enough to deliver to a mouse is one thing, but producing enough to deliver to humans is a huge hurdle in gene therapy. And that's because these viruses are actually very complex to make. They're quite expensive to make, and we need to make sure that they're safe enough to then deliver to patients. So this manufacturing issue is a massive hurdle that is faced by gene therapy scientists on the way to delivering to patients. And finally, of course, it has to be tested in many different clinical trials. So safety phase 1 trials, all the way up to trials testing if they actually work or not, which are the larger phase 2 and phase 3 trials. And these are all the many steps. And on average, this can take 15 years, this whole process. And it's no small feat to get from the beginning idea to finding the ideal product at the end. And just to manage expectations, that when we speak about gene therapy for TTP, we're very much in these early stages. And this is the sort of things that I was looking at in my PhD. So just to refer to a similar, a disease that gene therapy has successfully been applied to is haemophilia. So haemophilia is also a genetic disease, but also can be due to antibodies, and it's due to factor 8 and factor 9 deficiency. So just to highlight the framework or the timeline for haemophilia was in 1980s, they identified the genes that were responsible and were able to produce it in a lab. They then moved on to, in the 90s, to using recombinant protein therapy, so similar to the ADAMTS13 protein therapy. And this came about in the 90s. And in 2011 was the first successful clinical trial where a gene therapy for haemophilia was given to patients. And in recent years, we're about 10 years after the first patients were treated, and levels of factor 8 and factor 9 in their blood are still very high and still allowing for these patients to live normally without frequent use of protein therapy. And just to say that of late, the investment from pharma companies going into gene therapy for haemophilia is huge. So 450 million was the reach and purchase by a pharma company for a gene therapy product for haemophilia A. So now the pharma companies are getting interested, and what happens when they get interested is they get a lot of clever people to iron out the issues that I identified in the previous slide, such as manufacturing. And with the more investment they put into it, the hope that is that with time, these hurdles won't be as significant as they are now. So just to summarize, that gene therapy is a new exciting era of medicine. It's still in some diseases at very early stages, but in others, fully licensed gene therapy products are available. And I think gene therapy has the potential to treat a wide range of diseases, and I've begun to look at potentially applying gene therapy to TTP, with still a lot of work to do, but just at the early stages. And yeah, just to say thank you for listening, and I'm very happy to take questions. I hope I will be able to answer as much as I can. Obviously, with my science background, I'll do my best. But